Okay, good morning everyone. Welcome back, day 2. I see that we have late risers, so thank you for being here, the early risers. The opening remarks will be very short. Can I have the clicker, please? It's always the clicker. Okay, day 2. Just again, quickly overview of today's schedule. So first we're going to have the keynote, which is very exciting. And then the most important thing, the shuttle leaves today at 6:10, which is different from yesterday. So make sure you don't miss the shuttle. And again, we have various exciting talks and the breakout discussions, as well as the poster sessions later today as well for the second set of speakers. Okay, so that's it for today. I'm going to have Al come up and introduce the keynote speaker.
Thanks, Jean. Oh, sorry, Angela. Well, getting ahead of myself. Okay, so I'm very proud to introduce our keynote. It's Professor Jean Yang from Sydney. She's a leader in statistical bioinformatics, specialising in developing methods for molecular data in cutting-edge biomedical research. She's currently the director of the Sydney Precision Data Science at the University of Sydney. Her research has driven significant advances in integrating multi-layered biological data, including single cell, by reducing extraneous variability and addressing heterogeneity. So I'd very much like to introduce Jean. Thank you, Jean.
Thank you. And thank you. Oh, I think I'm just okay. Thank you so much for the organizers for inviting me. It's such a pleasure. I have a great time yesterday listening to all these fantastic talk. And what really strikes me is the multidisciplinary environment that we are all in. And so the talk today really is another exemplar of how many different disciplines could actually come together to actually work on something interesting. Okay, so before I begin, I just want to acknowledge that the work that is actually presented here today is done on the land where Sydney University is, the Gadigal people of the Eora Nation. I want to pay respect to elders past, present, and emerging. So, as Al said, I'm the director of the Sydney Precision Data Science Centre, and so just a quick introduction of our center. Basically, it's a collective of people, actually very much like people in your audience who are interested in solving challenges associated with modern biology and medicine. And so we come from many different disciplines. So there's wet lab people, but there's also computational people from computer science, applied mathematics, statistics, and so on. And so what I'm going to talk today is about getting the most out of spatial technology, given the amount of spatial work that I actually saw in the presentation. I think that needs no further introduction. But this slide is just one slide that was taken from a recent benchmarking of spatial deconvolution method by another group that sort of showcase the number of spatial platform that is actually out there. And so I'm going to touch on platform at 2 different resolutions today. One of them is the spot-based platform, which is actually a lot more common, and then the subcellular level technology, which is I would say, the cutting edge these days. Okay. So spatially resolved transcriptomics. So to begin with, I'm going to look at this particular platform where it allows you to look at spatial data, but you're looking at multiple cells per spot. So your measurement in itself, it's still not quite at single cell resolution, even though you get spatially resolved information. So the 2 complementary imaging modality that one can think of is, number 1, is this molecular imaging, right? So this spatially resolved transcriptomics information. We know that the strength of this, and that's why there's a lot of interest, and I think HCA Asia is talking about starting this spatial flagship program, and there's going to be discussion today, as well as we had discussion yesterday about spatial biology. So transcriptomic expression with the actual spatial content that provides a lot of information, but we all know it's relatively higher cost. Relative to what? Relative to standard H&E, which is the current standard for many diseases, so most cancer. It's what I call an accessible standard platform in clinical care at the moment. So can we actually get the most out of these two data? The question that people have been asking, and so there was a talk yesterday where the speaker got asked the question, can we actually predict gene expression from H&E alone? And I noticed there's actually two presentation out there in the poster yesterday also on this topic. So I just want to talk about our experience and our journey looking at this problem. Can we do this? Is this even possible? So back in 2020, there was a paper from James Zhao's group basically demonstrating this is not a blue sky idea. It's absolutely possible to actually get this information. And you may actually ask, why is this of interest in a translational setting? So there is many interests, and people like to do this, leveraging these two type of modality for many different type of question. I'm going to come in mainly because of the place I am from a fairly translational angle. And so imagine you have a lot of patient, individual, where the standard of care is this H&E platform. Imagine you can do a study design, limited sample, where you actually learn very deep the relationship between the H&E and the spatial gene expression data. Build some sort of model so that without measuring or without doing this experiment further on the existing platform, you could actually simply predict the spatial expression and utilizing both information together to actually build another layer of predictive modeling to actually distinguish between, say, good or bad outcome or responder or non-responder. So the concept can be used from a diagnostic setting. The context could also be used in a monitoring or predictive setting. Now, why is this? This is really trying to unlock the amount of histology data that is already out there. So recently there's a review article that actually shows, basically curate just breast cancer alone, all the breast histopathology H&E data that's actually available. So imagine these data could actually be utilized to look at spatial expression if this prediction was plausible. Then it comes to the really big question, right? What are the solutions out there and how good are these solutions? So with a lot of my work, rather than just starting to think which deep learning method I'm going to use, whether should I build a foundation model, I often like to start about benchmarking what is out there, knowing the strength and the limitation. So this was a little bit old already, and you can just see from 2022 and 2023, so I haven't added the number, the list is way too long. Basically, it's like a competing, the number of method that goes out there is scarily fast. Once the concept goes out, a whole bunch of method goes out there. And you can see the main architecture. Really, it goes from the CNN, transformer, anything that you can think of that's really in CS at the moment, it's probably not been applied. So we then selected about 10 of them. And I can tell you the truth is the ones that we managed to get it to work within 3 weeks to 4 weeks. Some, it was just beyond our capacity to get it to work. And so I can talk about the challenge of benchmarking, but this actually took us much longer than I anticipated when I first embarked on this project. So these are some of the earlier methods. And then what do people do when they look at this? So the most straightforward idea is if we can cross-validate it with ground truth data, so because they have measured H&E, we have ground truth data, remove the ground truth data, test it, compare, so you have the predicted number, you compare with the ground truth, it seems really straightforward, right? And so you're just looking at the correlation, and that's what most of the earlier papers actually have shown. Most of the validation compare the correlation of multiple methods. So this method, including the next one, here's another one, similar, they look at the Pearson correlation. Now, as a statistician, when I first look at it, I thought, wonderful, we can see which method is better than another. Then I realized, sorry, I'm just going to go back. I look at the y-axis. And the y-axis tells me that we are really looking at correlation of 0.1, 0.15, 0.05, and I ask myself, am I comfortable? Okay? So while one can actually say, I'm not comfortable, I don't believe in it, I thought a better, more constructive mentality is to actually say, maybe it's not the method, it's just the way we actually go about evaluating it. So I set about thinking, could we do this benchmarking design better? In addition to looking at whether we can actually reproduce the ground truth, what about translational potential? So let me sort of go through and talk about what we looked at. Obviously, we have to reproduce what people have done out there just to demonstrate, this is the same phenomenon, right? So we took the data that's published in most of these papers. So there's a HER2 breast cancer data as well as the skin cancer data, only on spatially resolved, spatial transcriptomics. So these are spot-based information. And there you go. The question is, if we repeat it, compare everything, you can compare and contrast all these various different methods. And most of them, the average correlation is about 0.2, and then you have to ask yourself, are you comfortable? Are we happy with it? So a small simulation that I actually do, I realized something that was taught to me when I was an undergraduate many years ago, was that correlation was something that's only meaningful if you're looking at signal. If you simulated a huge amount of noise, like 80% white noise with 20% signal, the correlation will drop. So this tells me that instead of looking at every single gene and looking at the average correlation, maybe I should just look at how well correlated is. What's the top average correlation, right? And the numbers is actually a lot higher. The top one usually is about 0.5 or 0.6. And then what's most important, the genes that they found out are really meaningful. So the top genes that in many of these methods found out, say FAS and MY12B, they are actually known to actually have relationship or associate with cancer progression, which tells me that, you know what, despite the average correlation, is probably not the metric to use, there is actually signal in this prediction. So while if you look at correlation, maybe it's more meaningful to assess this in terms of highly variable genes, differential expression, non-biomarker, etc. So that is one angle. The other angle is, remember at the beginning I say, why do we want to do this? We want to do this because we know whether it can predict on new samples. So here, I look at whether we can do it when I apply to TCGA data. So luckily TCGA data so I just decided because the data trained was from breast cancer, I look at TCGA breast cancer. And in there, we took around 600 samples, so there's actually 600 H&E. This takes a huge number of time to actually try all the different methods onto this data set. So for every single method, we estimate the TCGA output from those methods, and then I decided to keep it very simple. So at this point, you can say I could take spatial information in. We didn't do that. By the time you try to apply 600 things to 10 different methods, you think twice. The next step, we kept it quite easy. We simply linearized the whole thing, we built a Cox regression model, and we just look at survival. And what did we learn? So the typical evaluation is a C-index. The C-index, some of them, they're not great. So I think things can be tuned. So I didn't really tune this model much, but I think relative to many different methods, you could see that some of the C-index can reach 0.6, others can't. And you can also look at the survival difference for all the different methods. So every column is a different prediction method that's actually out there, and every row is a different cancer type. So we look at HER2 positive, we look at TNBC, and we also look at luminal. So what we say is obviously some kind of cancer is more challenging than others, right, as expected. But if you look at HER2, some of them you can actually see discrimination. And so this is looking at those different methods, their translational potential. And my conclusion was most of the method is able to discriminate at least one of those cancer subtype among these two risk groups. What this is highlighting is you can go in, and in this bioarchive paper that we have, we go in a little bit to talk about, how this is related to different architecture and so on. But the take-home message I got is we are not looking at white noise here. We're not looking blue sky research. Here, with better modeling, better thinking, there is actually signal. This is actually a plausible, a feasible pathway. And just to finish the benchmark component of, in this benchmark component, we look at, people look at computational efficiency. So the really two unusual design that we have is, number one, we put in the effort on the clinical translational impact. The other part is we follow one of the recent benchmarking paper to actually look at user accessibility. So for every single method that we tried, we examined many, many different aspects of user accessibility. So you could see, what strikes me is this is not a mosaic pattern. You either see black all across, or you see white all across. And that actually reflects just our community's behavior, which means we all do the same thing, but we are also missing the same thing. So what do we do really well? Everything is open source. Everything is out there. There's a package. Everyone can use it. What we don't do so well, and this is why I say every method took one of my students or two students about 3 weeks to get it working, is because it just doesn't run straight away. Because of, and I think this is at no fault of the any of the method development, I have to say we have the same issue for the product that we get out, is how many platforms do we actually get to test on? We don't. We test it on our own computer, our own system, and when system change, it's really hard to get it working. And there's just so many different type of package, so many dependency. There are big effort in bioinformatics community using Docker, using Singularity and many different systems to try to make things easier. But I think we still got a fairly long way to go in terms of that. As an example. So some of the method we actually have to go in to actually to tune it a little bit, because when we write method and our evaluation data set is small, 1 or 2, and this is especially true with spatial transcriptomic data, it's that at the beginning of a single biotechnology, this will happen. When this becomes more established, in about a year or 2 times, when there's a lot more data, then this will stabilize. But at the start of a technology, this is what happened. It takes a long time to actually get this sorted out. So very hard to get things to work. Some of things did, even though things are available. So I think this really tells us that availability of software, how do we actually ensure that quality assurance, and I'm just as guilty of this, it's something that I'm now keeping at the forefront of my mind a lot. So a summary of, before I even look at what we can do and our learning, we learned that, if we use a better correlation, so correlation on spatially variable genes and correlation on highly variable genes, you get much higher correlation. We look at that there is, if you apply it to an external data set, most of this method is able to discriminate high risk and low risk group. And most importantly, community effort really has to be put in to try to minimize overhead of entrance into a new area of method development. And to this end, just a little advertisement. Mark Robinson from Zurich and myself is actually running a modern benchmarking conference in Ascona next year. So if you're interested, or if you have people in your team who really want to get into this space for bioinformatics, QR code is there. Would really like to see many of you there. I think it needs a community to actually address this issue seriously. So what we also learned technically from that benchmarking is that many different architecture didn't really make a huge difference to the results at the survival outcome. So it makes me think, do I talk to another amazing computer scientist to actually, so I have a good discussion with my, with Professor Jim-Man Kim, my, an a deep learning imaging expert at Sydney University, my collaborator in this project. And we come to the conclusion is maybe we don't need a fancier method, but maybe we need to change the data input. So given that our benchmarking, we couldn't find a single method, even the ones with fancy transformer, that really outperforms the other, what could be another way you can actually improve is to change the learning data. So we decided not to look at spatial transcriptome, spot-based, but really utilize the subcellular spatial resolved data. So this could be the 10x Genome, the VisGen Merscope, NanoString Cosmic. These are the 3 platforms that we have experience with. So we have a look at that, where now the measurements, you have 150 transcript or so within each of the measurements. And we were able to get on top of this because, so I'm just going to get to the cellular level. We were able to get on top of this because earlier this year, we actually have a cell segmentation paper called BitCell, where we cell segment this utilizing biologically informed reference information. And so we could take most of these subcellular images, run BitCell through it to actually get the cell and then put that into our learning. And so what you are trying to now learn is the morphological relationship of a single cell with the expression of the single cell. So that learning makes the unit of the sample at the cell level rather than at the individual sample level. So GIST is our proposed method that actually takes H&E data, subcellular spatial transcriptomics, and together with a single cell reference data, so take 3 layers of information and actually use, pretty much a fully connected deep learning. So it wasn't a very fancy method, but the loss function takes into account of cell type information, it takes into account neighborhood composition, it takes into account of nucleus morphology, because that was actually a more stable morphology to look at, to actually predict both cell type and expression. So we simultaneously have an estimate of the cell type and the expression in our module. And so here's the results of our prediction. So here you could see that using the Xenium breast cancer data, we were able to look at the ground truth, look at the predicted, and if you just look at the cell type composition level, you can say that you can mimic the cell type reasonably well. We tried it in the lung. Again, this is the 10x Xenium data. Look at the lung data, and I'm just changing the way we presented it. So ground truth proportion with predicted proportion, it's fairly the concordance was pretty good for lung, and the concordance again was pretty good for melanoma. But this is not quite at the single cell expression level, right? So this is just getting to cell type. So this tells us we are on the right track. Now moving to the genes, we can again now look at the Pearson correlation. And so at this point, we only look at spatial variable genes. We don't look at everything because we want to look at the signal. So in the spatial variable genes, we also highlight the fact that if you look at the top 20 spatial variable genes, you get that signal. And here, the correlation could be as high as 0.6 or 0.7. Really, the predicted expression for genes of interest is really there. And just to make sure that we are not predicting everything, with some sort of just copying the data across, you could see that the non-variable genes, there is no correlation. And that's what we want. We're just trying to show that you're getting correlation with signal, and of course not quite 0, but it's very low. And that's saying that for the white noise, for the genes that we don't expect, we are not really, we just the estimation of expression is fairly random. Okay? And I think that discrimination is actually really important when we're starting to look at this ground truth of H&E to spatial gene expression prediction. So this gives us confidence that we're actually getting that information. And also, we went back and look at, can this be flexible enough to look at spot-based information, the algorithm? And so it shows that it can, and be in the spot base because we were looking at the PCC, unfortunately, not the best metric, but it just say that it sort of works. We can perform just as well as all the other methods out there. So it runs. So what I'm actually going to spend the other time on is how we are actually using this data, assuming that we can predict. So if you want to find more testing and validation and all the technical details, there's actually a bioarchive on GIST that's actually out there. And I have to say, this project really highlights the potential of working together with a deep learning computer scientist with imaging expertise, as well as a pathologist and a breast cancer pathologist who kindly actually check a lot of these pathology images for us when we are utilizing this. Okay, so this is the detail. So in the next 10 minutes, that's the time that I have, I want to talk about the potential of this data set. What can we do with it? And maybe prompt everyone to think about what are the methodological development that one can use if I can actually share with you this kind of predicted data, and how can this be utilized in your next study design. So what are some of the analytics that is actually plausible? So what we really have created for the TCGA is an in silico platform. Currently, TCGA have H&E data, they have RNA-seq data, have proteomics, microRNA, so there's multiple platforms. And so while we didn't actually take any samples to perform a wet lab experiment, by doing this prediction, and we've done about 400 of these on our data set, you are creating one layered of platform that allows you to learn a little bit more. Now, I just want to clarify, this is not as good as if you've done a real wet lab experiment, but it's a lot cheaper, right? So that's your computational cost. And this could be an initial set for you to decide what samples to do the actual wet lab experiment, because you can computationally predict it and you can know which samples are important. So I'm hoping this could actually form an important design consideration going forward with spatial transcriptomics. So we've introduced one layer, so this new layer of in silico platform for the TCGA data. I'm only showing it 92 samples, which is HER2 positive at the moment. And so obviously this is, you can deconvolute because we have estimated the expression. You can deconvolute the data just like you can take the RNA-seq data to deconvolute it. And then you can ask a lot of questions of interest. So we examine molecular signal from two groups. So say ER positive, PR positive, and ER negative, PR negative. And we look at cell type specific differences, so it's different state genes. So you can actually see that here, you can look for certain cell type. So this is your typical DE analysis that you can do now. At the cell type level, you could also find new sub-cohort. So when I was looking at this data set, I actually discussed with Yue, so my postdoc who actually did this work, and she's here in the audience today. So we look at the ER positive and the PR positive group, and I said, there seems to be a lot of pattern. Could I cluster and actually define the two patterns? And you can see that if you cluster, for some of the sub-cell type cohort, there seems to be a different pattern, right? So you can discover that sub-cohort, and then that brings us to, can I extract more information? So for an individual, imagine you have an H&E, you can predict this outcome. You could also try ways to extract features. So here I extract features just on gene expression. I can also extract spatial metric, such as what are the relationship between the different cell types. And this is why I talk about we might be interested in the actual marker level, but more importantly, it's just really the cell type information that we want to get through, because we can look at patterns of cell type. Are they intermixed? Are they not intermixed? So there's a lot of spatial statistics out there that initially was developed in the geoscience space that we can borrow and use, Moran-I, L statistics, to actually measure segregation, right? That's example to the left, or integration or mixing for two different cell types. And so with that information, you can actually measure out of all these cell types in this predicted, are we seeing a lot of mixing or not? And therefore, with that, you can define, using this pattern, differential patterning, and we can see, can this differential patterning make a difference between these two sub-cohorts? So imagine I'm interested in cluster 1 and cluster 2, these two sub-cohorts that I found out with different expression. Is there a difference in their patterning? And using discrimination as a metric, we can see that, yes, we can find the patterning not only in the expression, but there seems to be a difference also in the pattern. So I would take one to talk about utilizing this very classic five-fold cross-validation to actually see how can that actually add translational power. So most of the time, when you look at any model, people tell you the overall accuracy. So what we can introduce here is individual accuracy for the individual people, using a very simple analytical trick. So imagine you have data, your genes and your patient. So I just want to talk through this concept. You learn and you train. This is what we teach in computer science and in statistics. So five-fold cross-validation gets you an outcome for one of them. Imagine you repeated this 100 times. So now you have a matrix of the performance of all this learning and training repeat. Traditionally, we will get an estimate for every repeat. So we say on average, the performance is 0.8 accuracy plus and minus something. This is what you see every time you look at risk prediction. But if you don't take the average and you just look at this data in a different angle, do you think all samples is classifiable equally well? So I think most people can start seeing that if I just take the the column sum, rather than the row sum, I could say among the 100 repeat, how do individual person perform? 100 out of 100, I always get it right. So it doesn't really matter what model, even if it's an imperfect model, I get it right. 50 out of 100 or 0 out of 100. So this actually allows us to start unpacking what works well for what individual. Because for me, it allows us to actually ask this question: does spatial transcriptomics add value? So here is the one where we look at, if I build a model with traditional overall markers, your microarray, your RNA-seq. Number 3. I also look at if I use cell type specific expression. This is what you can get when you have scRNA-seq. You could also get cell type proportion when you get scRNA-seq. But I could also find spatial marker, which is number 4. So I'm looking at what happened if I look at, traditional markers and new markers. Now, notice that overall, if you actually run an average of these 4 things, it's next to impossible for spatial markers to outperform mutation-based information, because that is what the population have. So 2 ways to look at this problem is sometimes we look at a sub-cohort of individual where mutation markers of risk prediction don't work for them, and we see whether there's another solution. But this precisely illustrates this information. So every row is the individual prediction for a different method, right? So for every column is an individual. So when you see the really dark red and the really dark blue, it says I'm getting it right. Whether it's a good outcome or poor outcome, actually in this case, is cluster 1 of cluster 2. Whether it's cluster 1 of cluster 2, I got it right. But if it's white, it means I'm getting it wrong all the time. So majority of the individual, it really doesn't matter what platform you use, you're doing really well. But there's always some individual that it works for some platforms. So if the ones that you look at, you can see the cell type specific expression actually does really well where the traditional actually fails. It means that for this individual, they need something more. And here, it shows that for 1 or 2 individual, the spatial marker makes a difference where traditional overall expression and single-cell RNA-seq actually don't get there. So this is just a way to sort of unpack that different proportion of the population actually will benefit from different amount of information that we are getting from histology. Okay, so I'm about to go into the last illustration of what we can actually use. So far, I've been looking at the predicted expression and what can you learn. And based on this learning, you can design a better spatial transcriptomics experiment going forward. But because we applied to TCGA as an in silico platform, it also offered the opportunity for us to actually combine and look at this with other modality in TCGA. So here's an example of modality that we're going to look at. So we're going to look at the genome data. So what I've done is we look at copy number aberration. So imagine for every single gene, I examine which are the individual which have really CNA gain. And I also look at the individual with CNA loss. So this concept is I repartition the individual based on the gain and loss of CNA. And then I ask, when I compare the patterning of these 2 cohorts for every single gene, do I see a difference? Do I see a lot of differential patterning? And so this differential patterning is defined by nearest neighbor correlation. So this is what I've done earlier. So I look at patterns, whether they're intermixed or they're segregated. So now I look at different cohort and I say, do this cohort as a group has a lot of intermixing, or do this cohort as a group are very well segregated, and what is their relationship? And so then you can scan it through genome-wide information, and what you can find is there are certain regions of the genome where we find a lot of differential patterning. And there's certain region, and so one of this area is in chromosome 17. This happens to be the HER2 plus region, and in another one is this 8q24. So there is like a little region where I call maybe differential patterning hotspot that is related to CNA, and we actually found an existing paper, that actually found that, for breast cancer in this region, there is actually a lot of cancer susceptibility cohort. And so you could say that this is a way to actually link sort of genetic and other somatic mutation information with differential patterning, seeing whether there is actually heritable information coming through. And I'm trying to get into genetics, so if there's genetics people in the audience who are finding this kind of thinking interesting, would love to have a further conversation and collaborate. So just to wrap up, at the start of my talk, I talk about the importance of benchmarking and how that benchmark actually guide how we actually approach our methodological development. And I introduced GIST. Basically, it's a multimodality deep learning approach to predict cell type and as well cell level expression from H&E image, especially taking advantage of high resolution data, so subcellular data. Our team at Sydney not only work on GIST, there's a whole bunch of other single cell related and spatial related packages in R that we produce. I also utilized the last 10 minutes to actually discuss computational derived resource that we could actually create to actually guide modeling and methodological development if you are a computational person, but I'm also hoping that some of this initial result could actually guide experimental design and studies. And we try to demonstrate there is a potential for translation in precision medicine for this. Now, this work couldn't be possible with a team of people at Sydney Precision Data Science Centre, the support from various funding agencies. Just to highlight, the computer science collaborator, Professor Jimen's Kim team and his team, as well as the Center for Cancer Research and the Breast Cancer Institute for guiding the breast cancer component of the work. Thank you very much for your attention.
Thank you, Jean. Have we got any questions? Yep, we've got one at the back there. From Drew. Keep your hand up. Okay, Drew first. Drew first. Go ahead.
Hi, Jean. Great talk. I was really interested in the inter-individual variation that you see across these different modalities, and where you see certain individuals that you can gain information from to help predict their responses, if I'm remembering correctly.
Yep.
I'm wondering if there's an easy way to identify which individuals those additional modalities will work for, or if that's going to be more challenging.
No, it's a really good question. And so since in some of our previous melanoma work, if you have this, how well an individual perform, you can imagine that could be your Y variable, and you can regress it against any clinical data set. And so it's just changing the problem, but you can actually determine what makes someone works. And so for that, we in the previous work, we have highlighted, okay, if someone have tumor extra nodal spread, then they work better or not. So you could actually identify other things, and that's precisely, even apart from looking at how well it works, you can use that value as a Y variable and do further analysis to identify exactly what you're interested in.
One over there.
Hi, Jean. Great talk. Thank you. Yeah, I have a question regarding when you took top X gene from the prediction, because those genes have more signal than they have noise. I'm wondering if you have tested across different runs of the same model or different models. Are the top X genes consistent across all these different runs? And
so I can answer that. The top X genes the ranking will not be identical, but if you have they are relatively highly overlapped. Does that make sense? So it's not always the same. There's always things that works in one and not in the other, but they are approximately the same. And so you may ask, do you do top 20, top 30, top 50? The truth is that correlation would just drop as you go down.
so I'm wondering for the genes that are always not predicted well, let's say usually the immune type, how do you propose we try to predict them better?
So my question is, I don't think they're not predicted well. So I think you're asking a orthogonal question, is whether the gene is actually predicted well versus I don't expect prediction for those genes. What I'm trying to say is I don't expect correct prediction for noise. So if you have an experiment for 300 genes, but there's only 100 signal, I don't expect those genes which are not expressed or they're not really doing anything to actually be able to predict. You don't want to show your method can predict them. So a better way to actually look at that would be saying that, okay, not the top 20, but maybe 50 to 100 or something where you expect to have signal, but they're not predicted well, how we can improve upon that. So I would say that if you look at the correlation drop in your case, I would expect a sharper drop from a better method than a more gradient descent method. And that would be the metric I would use to answer your question. A good suggestion.
And if I'm allowed, one more little question. Just for, okay. Have you looked at bulk? Bulk prediction, thank you.
I don't quite follow that question, but maybe we can. I'm happy. I'm around the whole day, so happy to talk to anyone afterwards.
Yeah, hi, super nice talk. I just have a question. So in the first part of your talk, actually, you demonstrate training not using like spot level spatial data, but instead like the subcellular. But I still do not get it. So do you think using the subcellular data for training is better or not? Because you compared your model with other like spot level data, and but just demonstrate is as good, but not really much better, right?
Okay.
So,
I just want to clarify, I do in our own, when we for the same data set on TCGA, when you learn from spot versus when you learn from SST, subcellular is better. There's no question. Sharper correlation and everything. And my reason for that is because your learning is cleaner. You're learning from one cell's morphology to one expression. You are not learning from a mixture of information. There's way too many combinations in that mixture, and it's much harder to unpack that mixture. That's my
Thank you so much.
intuition. Yeah.
Okay, we've got time for one very short question. You sure?
This is a very provocative question. It's a great talk. Do you think one day we'll be able to just go from H&Es straight up to the diagnosis or outcome, sort of like bypassing, the other information in between? Because a lot of those genes don't
This is not this is a fantastic question. I think this is the ultimate aim. What you do inside, whether you actually estimate or not, I think that's actually irrelevant. From what people were doing, they would be looking at the H&E, they would just average. So the way I think the conceptual innovation here is not that I predict spatial expression, is that I leverage the information from spatial expression to allow me a better prediction at the end.
Okay, so with that, I'd like to thank Jean. And I'm sure if you've got any extra questions, she'd love to talk to you at morning tea. So now we're going to move straight on to the next session, and I'll introduce Ralph George, who's one of the co-chairs.
Yeah, good morning. So my name is Ralph Jauch, here from the University of Hong Kong, and it's my first HCA. I'm really happy to be here to co-chair the session with one of the founding fathers, Jay Shin. So this is the neuroscience session, and we are going to have a disease focus so that diseases will be increasingly important in future HCA's initiatives. So we will have two talks about, one talk about cancer and two talks about neurodegeneration. And our first speaker is Hiromichi Suzuki from the National Cancer Center in Tokyo. So he's very prolific. He has done a lot of influential work on various types of cancer, and he will share his latest research about brain cancer. Looking forward to your talk.
Thank you for a kind introduction. So I'm very pleased to have this opportunity to present our work today. So I have a cough, so I'm afraid that you may not be able to hear me clearly. So today I'd like to talk about sequencing analysis of brain tumor, focusing on cell of origin and genetic alterations. So this session is neuroscience and neurobiology, but my talk is also about cancers. So I will present our findings, which utilize single-cell sequencing data. Okay. Oh, it doesn't work. No? Okay, now works. So I have no conflict of interest. Oh. Okay. Okay, so brain tumors are classified into more than 130 distinct types. So, Okay, so with such a large number of disease types, so clinical diagnosis and research are a little bit challenging because of its complexity and also the limitation of available tissues. So recently, it has been reported that brain tumors can be classified based on DNA methylation pattern clearly. So DNA methylation changes is a combination of somatically acquired changes and characteristics which reflect the cell of origin. So the diversity of disease types of brain tumors suggests the existence of the diversity of the cell of origin of brain tumors. So brain is the organ with the highest diversity of cell types in the human body. So the brain contains approximately 3,000 different types of cells, each of which has distinct characteristics and functions. So specific genetic alterations must occur for malignant transformation. So for example, mutation in gene X can transform cell A into cancer. On the other hand, mutation in gene Y can transform cell B into cancer. So, however, different combinations do not lead to cancer. So, mutation in gene Y cannot transform cell A into cancer. So, specific genetic alterations are required for the initiation of cancer. So therefore, identifying the cell of origin and the genetic alterations is very important for understanding the pathogenesis of cancer. So for this purpose, so single-cell sequence is a useful approach. So cancer exhibit intratumor heterogeneity. So single-cell analysis enables us elucidates the characteristics of individual cancer cells. Cancer also exhibit hierarchical clonal architecture, so single-cell analysis can evaluate differentiation and progression stages. So estimating the cell of origin requires a reference catalog of normal cells. So by comparing characteristics of cancer cells and the reference catalog, so we can estimate the cell of origin. So my talk is about medulloblastoma, so which is the most common malignant pediatric brain tumors. So recent genetic studies reveal that medulloblastoma comprises 4 molecular subgroups: Wnt, sonic hedgehog, group 3, and group 4 with their own distinct clinical and molecular features. So medulloblastoma is a still lethal disease, and survivors often suffer from severe cognitive disabilities resulting from their treatment course. So therefore, new and effective therapies are still needed. So I will explain medulloblastoma subgroup briefly. So Wnt medulloblastoma is characterized by the Wnt pathway activation, so usually caused by CTNNB1 mutations or APC mutations. So Wnt medulloblastoma is associated with better prognosis, and the sonic hedgehog medulloblastoma exhibits the activation of sonic hedgehog signaling pathway caused by genetic alteration in the sonic hedgehog signaling pathway, such as PATCH1, SMO, SUFU, or GLI2. So group 3 and group 4 medulloblastoma has genetic alterations in MYC, OTX2, and PRDM6. So, however, many group 3 and group 4 cases lack driver mutation. So, group 3 and group 4 medulloblastoma, their detailed mechanisms of tumorigenesis is not well understood. So group 3 and group 4 medulloblastoma has very similar pattern based on DNA methylation or gene expression patterns. So actually, some of group 3 and group 4 cases lie at the boundary between group 3 and group 4. So Wnt and sonic hedgehog medioblastomas are well characterized. So Wnt medioblastoma is believed to arise from mostly fiber neuron in lower limbic loop with the abnormality in the Wnt signaling pathway. On the other hand, sonic hedgehog medioblastoma is thought to arise from granule cell precursor cells with the activation of sonic hedgehog signaling pathway. So those results are obtained with mouse developing brain data. So on the on the other hand, we don't know much about group 3 and group 4. So in group 3 and group 4 medioblastoma, the most common genetic abnormality is PRDM6 overexpression. So this is caused by enhancer hijacking. So by the tandem duplication of SYNCAP blockers, its enhancer locate near PRDM6, result in overexpression of PRDM6. So however, how this activation contributes to tumor genesis is not well understood. Okay, so to reveal further pathogenesis of group 3 and group 4 medioblastoma, so we analyzed RNA-seq data with a large cohort. So we called mutation with RNA-seq. So this is a little bit challenging. So because RNA-seq data has no matched control, and also there is RNA editing. So we developed our pipeline for mutation calling. But today I will not talk about the detail of the mutation calling. Probably many of you are not interested, interested in bulk RNA-seq analysis, but if some of you would like to know more detail, please ask me later. So as the result of mutation calling, so we discovered a novel mutation in CBFA32. So this mutation is truncated mutation, so most of mutations are frame shift mutations or nonsense mutations. And also missense mutations are enriched in NHR1 domain. So NHR1 domain interact with PR domain of PRDM family. So PRDM6 is one of the PRDM family gene. Also PRDM6 has PR domain. So interestingly, cases with CBFA32 mutation has normal expression of PRDM6. Sorry. So with a further investigation, we noticed cases without CBFA32 mutation or PRDM6 overexpression has recurrent deletion on 16q by the SNP array copy number analysis. So the overlapped region contains CBFA33 gene, so which is another family gene of CBFA. So CBFA32 is not reported as cancer driver gene. So we investigated interacting proteins with CBFA32 using in vivo experiment. So among the interacting proteins, so we found KDM6A. So KDM6A mutation is also recurrently mutated in group 4 medioblastoma. So the enrichment test of interacting protein revealed epigenetic modifications and hindbrain development. So this data suggests CBFA32 probably works on brain development. So if we see the mutation in CBFA complex, so PRDM6, CBFA22, 23, KDM6A, those mutations are almost mutually exclusive. So and this data suggests the disruption of CBFA complex failed hindbrain development. So now, so we have the data of genetic alteration. So then we would like to identify the cell of origin. So the first, we investigated the expression of CBFA2T2 and 2T3 in human developing brain. So we detected the expression in the rhombic ellipse subventricular zone indicated by a red triangle. Oh, thank you. So, okay. So now, we utilize single-cell sequencing data. So we compared medulloblastoma data and human developing brain. So we detected the cell of origin. So the green indicated group for medulloblastoma, and the yellow indicate group 3 medulloblastoma. So group 4 medulloblastoma resemble neural progenitor in rhombic ellipse subventricular zone, and also early unipolar brush cells. And group 3 medulloblastoma resembles more immature neural progenitors. So now, so we have genetic alterations and cell of origin. So these findings enable us to conduct functional analysis. So I will skip the detail of in vitro experiment. So the result discovered, so CBFA complex mutation disrupt normal cell differentiation, leading to cell rest, and those cell rest is developed to medulloblastoma. So through the various efforts of researchers, now we know much about the association between genetic alterations and the cell of origin of medulloblastoma. So these findings lead to some development in basic research and development of novel therapies. So maybe we can target the vulnerability of the cell of origin and or force the differentiation. And also the prophylactic treatment can be expected, but the development of diagnostic test is necessary, but if we detected those regions before developing to cancer, we can remove those lesions. And also now we know the cell of origin and genetic alteration, we can establish mass disease models. So this is a conclusion. So medulloblastoma arises from specific mutations occurring in precursor cells, inhibiting their normal differentiation. So we discovered the CBFA complex mutations which disrupt cell differentiation, causing cell rest, which consequently develops into group 3 and group 4 medulloblastoma. So our research is supported by many researchers and also patients and patient families. Thank you for your attention. I'm happy to take questions.
Thank you for a wonderful talk. I know you had to fight with your cough, so thanks for still coming to us and presenting your exciting work. So apparently, maybe I start with the first question. You had to use a lot of advanced single-cell technologies to find the cell of origin. Yeah. So is this involving like lineage tracing technology, or does this involve some lineage tracing that you needed the lineage mapping to find the original cell?
Yes. But actually, cancer exhibit a bit different expression pattern from reference catalog. So because of this, so first we had detected this expression in rhombic ellipse. So we compared this lineage and cancer cell data.
Any more questions?
Really nice talk, thank you. I'm curious whether you think medulloblastoma could be replicated in an organoid model, and whether these kinds of cell of origin studies could also be replicated in such a model.
Sorry, I'm afraid that I have no idea about the organoid model. But so now, the important thing is group 3 and group 4. The cell of origin of group 3 and group 4 medulloblastoma can be detected with only human developing cells. So medulloblastoma is specific to a human. Maybe so we cannot establish mouse. Yeah. So this is very important progress. Maybe we have to use organoid or iPS and to develop the disease model.
So we would have time for 1 or 2 more questions. Well, if there's no more question, let's thank Hiromichi again for a very nice presentation. And I'm handing over to Jay.
Thanks, Ralph. Good morning. It's my honor to invite our next speaker, Junil Kim. He's an assistant professor at Sungil University in South Korea. And his research interest is in development of algorithms. And he developed key tools such as Tenet and GRN reconstructor. So yeah, we look forward to his talk on single-nucleus and spatial transcriptomics. Please.
Thank you for your kind introduction, and it's a great honor to present, have an opportunity to present in this HCA meeting. Yeah, I'm from Sungil University. Maybe you never heard about university name. Is there anyone heard about Sungil University? Thank you. So yeah, most of you never heard about Sungil University, so I want to introduce my university. I'm from South Korea. It's located in Sungil University, located in Seoul, and don't have any pointer. No. Anyway, so that statue is the symbol of our university, the white horse statue, and you can see there is a missing part in this triangle. So let me introduce the history of Sungil University. There is some reason why there is a missing part. So Sungil University founded on October 10, 1897, 2 decades ago, and in Pyongyang. Now it's in North Korea. Anyway, so at that time our country is we have one country, and it's founded by William Baird, a USA missionary, and on March 4th, 1938, the school closed itself because of, I can say, imperialism, and you may can find that in Wikipedia. Anyway, so in April 1954, Sungil was re-established in Seoul. That's why there is a missing part. So we wanna go further for the good university. And our department of bioinformatics opened in 2001. It's first time bioinformatics department in undergraduate course in South Korea, and I was the first student in that course, and I came back to my the same department. Okay, I think it's enough to introduce my university. I'm a bioinformatician, and I'm mainly try to develop tool for analyzing single-cell RNA-seq or spatial transcriptomics. So that 3 topic is I want to summarize what we can do using single-cell or spatial transcriptomics. I want to summarize in that 3 topics. First one is heterogeneity. We all want to know what kind of cells in our sample. And second topic is dynamics. So because we can obtain some trajectory, cellular trajectory, we can study dynamics. Third topic is cell-cell interactions. And today I want to focus on the second topic, cellular dynamics. If you want to hear another topic, please invite me again. And so using the, let's say we have dynamics. Let's say we have a cellular trajectory, for example, developmental path trajectory or disease progression. We can study, because we have time, which gene expression is cause, which gene expression is effect. So that's what I study when I was a postdoc. And so let me introduce that topic. So I developed TANET. So it's based on the pseudo-time ordered single-cell RNA sequencing data. We borrowed the concept of transfer entropy, which can calculate the causal relationship between two variables. So let's say we have a gene A and gene B expression along with the time point, then we can calculate the causal relationship using this transfer entropy. You may never heard about the TANET tool, but you may heard about this the cinema name TANET. So happened to be it was same. Believe me, when I name it, I didn't realize there will be a movie. But anyway, so there is a good thing. The name is same. I can make you memorize my name, the tool name. But bad thing is you cannot find my tool in the first page of the Google when you type the TANET. But you can anyway search my tool using TANET and single cell like that. So as I said, so using the trajectory, I calculate the causal relationship between genes. Then it means I can obtain the gene regulatory network. So I applied it to this mouse embryonic stem cell, and I prioritize in the gene network. I prioritize the transcription factor based on the number of targets. Then I can find the top 4 transcription factor is the Yamanaka factor, and also the famous transcription factor for the neuroprogenitor differentiation. And I compare the performance to identify critical regulator to other tool. Then only my tool can find that Yamanaka factor at that time. But recently, when I run again, they also can find that Yamanaka factor. But anyway, so my tool can identify critical factors on that trajectory. So. Okay, so TANET is very good for the identifying critical regulator. So I applied many different cellular trajectory, stem cell differentiation trajectory, and autophagy trajectory, and also hepatocellular carcinoma trajectory, and I can obtain a transfer factor list. And I validated using cell experiment, so it will work for the many different kind of different trajectory, cellular trajectory. So let me switch gear to today's main topic, Parkinson disease. So I want to apply my trajectory method, the gene network method to Parkinson disease progression. So we obtain, collaborate with Seoul National University, Professor Sung-Hae Park and Professor Jae-kyung Won and Dr. Suyeon Yu. We can obtain the single-nucleus data and vision data, and my two great students analyze the data, and as you can see in the figure A, so we first diagonized the samples in the brain bank of Seoul National University. So we obtained we diagonal sample to control and Parkinson's disease. As you can see in the H&E image, there is a black pigmented cells in the control, but there's not many black pigmented cells in the PD. I skipped that part. So that H&E data is from the midbrain and substantia nigra. So substantia nigra means there's black pigmented. And we also checked in the PD sample, when we stain the sample with alpha-synuclein, we found there is a Lewy body structure. So we diagnosed each sample, and we run the single-nucleus RNA-seq and visium data. So as you can see in figure B, we obtain from the single nucleus, we profile all the cells, and we annotate cells like microglia or astrocyte or oligodendrocyte, and neuron cell is very small set of cells, and OPC and endocellular cell, every cell, I can annotate with the marker genes, as you can see in figure F. And figure C shows it well integrated, so there is not that many difference when I try to integrate them, and each marker genes are well expressed in each clusters, I mean each cell types of the both of the control and PD samples, and there's not that many changes of the proportion of global cell types of global cell type like OPC, oligodendrocyte, and any other cell types. So yeah, we obtain single-nucleus RNA-seq, and so even if it's well integrated and show the clear cell type markers, there could be also changed transcriptional alterations within each cell type. So we run the differential expression analysis, and we found astrocyte and microglia have many differentially expressed genes, and it have also its own distinct gene ontology and pathways in this differential expression of astrocyte and microglia. So I'm talking about the trajectory. So I run the monocle, and I found the trajectory, and it shows the PD progression. As you can see in the lower figure of the monocle representation, you can see there is two trajectory which have a highly populated with PD samples. So anyway, it shows some trajectory from normal astrocyte to the PD astrocyte. So when we try to analyze the trajectory associated genes, we found highly expressed in the PD trajectory have a gene signature associated with the reactive astrocyte, like regulation of amyloid beta formation, like that. So, and we found some trajectory, and we also can find the gene marker genes, which shows highly expressed in the PD samples, and we check it using RNA scope. As you can see in the middle figure, there is many highly expressed of the CHI3L1, which we found in the trajectory analysis, and we also found the CD44 also highly expressed in the preclinical PD and also PD samples, but it's not that much high expressed in the control samples. And we also focused on the microglia. As I said, we focused on astrocyte and microglia, and the second one is microglia. We also found three trajectory, 1 to 3, and out of 3 trajectory 1 and 2 was associated with the PD progression. It have a many population of from PD samples, and we also run the same analysis. We try to find the trajectory associated genes, gene signature, and in gene set 2 was highly associated with this interferon gamma response signaling and JAK-STAT signaling, which is associated with the inflammation. So we also check the trajectory associated genes. We check one gene, CD163, is highly expressed in preclinical PD and PD samples in the microglia. The CSF1R is a microglia marker, and the yellow spot means the cell which have both expression of CD163 and CSF1R, which means in the microglia, in the preclinical PD and PD sample have a high expression of the CD163, but it was not expressed in the control sample. Okay, so that was our trajectory analysis, and I run TANET, and I found every trajectory I run, each trajectory I run TANET. I combine the genetic network, and I analyze the modularity in the network, and I found the green and yellow is associated with the astrocyte trajectory, and blue and red is associated with the microglia trajectory. And I found I only show the transcript factor in this case, there is in this figure there's no target genes, but every transcript factors are well associated in my TANET network, and I found four different modules, and each module have its own associated gene ontology or the KAG pathway. So I want to move on to another topic like the vision data. So until now, I haven't shown any spatial any vision data, but we only have a two slide of vision from a PD sample and two different regions of the same sample, and one is a medial and the other one is a lateral. And I just analyzed the simple clustering, and I found some the regional section of the spots in the clustering analysis, and the right figure shows the markers from the single-nucleus RNA-seq, and when I project it in the spatial cluster, it shows the cell type clusters are mixed up because it's not a single cell resolution, but each cluster shows a different expression profile. For example, cluster 7 and 8 shows high expression of the neuron, and it located in the upper region of the substantia nigra. It's not associated with the PD progression. So, my collaborator get back to the textbook of anatomy, and the pathologist found that this part is also each region, each cluster region is associated with each anatomical region, like a substantia nigra or VTA. I'm not pathologist, so anyway, so VTA or R region, that something like that. And because I want to focus on the trajectory, we have a trajectory for the astrocyte and microglia. I want to apply it to the vision data, and so I deconvolute vision data using a cell location, and I found so I have a subcluster in the trajectory from AC1 to AC8, and AC1 and 3 is associated with the normal case, and the other subcluster is associated with the PD case, and I found the normal associated astrocytes are spatially correlated, and the other PD associated subcluster proportion is also spatially correlated in the Visium data, as you can see in here. So the green part, the AC2, 4, 6, 7, 8 shows they are highly expressed in the substantia nigra, and the other AC1 and 3, the normal associated astrocytes are highly expressed in the other region, which is not associated with the PD. Like there's no in the substantia nigra, there's many deaths of a neuron cell, like dopaminergic neuron, but the other part there's still many live cells in the other region. So the trajectory associated subclusters are also spatially correlated in the Visium data, and it shows the trajectory. I don't have a normal case, but in this case, I have a substantia nigra, and the other region, each region shows different proportion of these subclusters. Okay, so that's end of my presentation. I don't have any conclusion. I want to quickly. Anyway, so and thank you for my group. This is my students. So we have many teams for tool development or disease single-cell RNA-seq or Visium, the spatial data analysis. And I thank you for my collaborator in Denmark, USA, and especially for the Seoul National University Hospital pathology department. Thank you, and I'm happy to take any question.
Thank you, Junil. Ralph?
Fascinating talk. So currently there are many cell therapy trials for Parkinson's disease using stem cell-derived dopaminergic neurons. From your talk, I understand what's going wrong in PD. The pathogenesis is mainly caused by microglia and astrocytes. So I'm just wondering if actually, if you're putting in these neurons and you still have all these rogue astrocytes and microglia, what's your opinion about, the promise and the vision of these cell therapy trials?
Yeah, it's a good question, and I'm not sure I can answer well. I'm not the clinician, but anyway, so what I found is not cause. We don't have any clue for astrocyte or microglia cause, but what I found is that there is many alteration in substantia nigra region of the astrocyte, and also in the single cell and vision data. So maybe we can target the trajectory associated genes in astrocyte and microglia, and it could have some role in the inflammation or other biological pathways associated with the neuron cells.
Any other questions? There's one back there and one in the front. Go ahead, in the back.
Right, hi. Just got a question for you. So you have loads of data. So what's the conclusion from all of the data then? So you said, and what's the advantage for your treasury compared with the one we are using every day? So I didn't get information from your talk. Thank you.
Yeah, what you found is alterated genes in the astrocyte and microglia. And we also compare it with other public data, and they also found there's many alteration in microglia and astrocyte. It's actually same conclusion, but I think we analyze it in South Korean people, and we found the same conclusion, and also we collect very high quality samples and based on the pathologist view. So I think it's a very good resource to identify the marker genes for the PD progression.
One quick question.
Okay, so in your single cell UMAP data, you said there's a few neurons, not so much. I'm wondering this is due to the isolation or because of the neurodegeneration. And did you see that in your spatial map? So did you also see that there are less neuron? Because theoretically there should be like 20 or 30% neuron exist there.
Yeah, it may have a both problem of the collection, and also we only collect the substantia nigra. There's many deaths of neuron cells. Yeah, that's the second region, and when you but vision data include not only the substantia nigra region. So the other region have a high proportion of the neuron cell, but in the substantia nigra have a less proportion of the neuron cell. I think this concordance with my single nucleus analysis.
Okay, but also probably due to the neurodegeneration in substantia nigra, right?
Yes, yes.
Yeah. Okay, thank you.
Maybe quick question on, for the sake of HCA Asia community, how accessible are these tissue banks for brain samples?
I didn't prepare the answer, but anyway, so I can say Seoul National University Hospital have many samples for Parkinson disease and Alzheimer's disease. I think we have lots of resource, but lack of grant. So, yeah.
Okay, great, thank you.
It's my pleasure to introduce the last speaker of the session, Jimmy Lee, who's actually a local hero. He got his degree from the University of Hong Kong, and he's now working at a Sanger in the UK. And we will stay with neurodegeneration, but switching from Parkinson's disease to ALS.
So Thank you very much. First of all, I would like to take this opportunity to thank the organizing committee for giving me a chance to give a talk here because as introduced, the kind introduction, it gives me an additional layer of talking in a place where I grew up. And also I would like to thank everyone for putting together such a great conference. So I'm a postdoc from the Omer Bayraktar lab, where we're interested in using spatial transcriptomic and multi-omic platforms to basically study the molecular and cellular nature in neurological disorder. And today I'm very happy to talk about how we try to identify shared molecular vulnerability in human neurons, in amyotrophic lateral sclerosis, also known as ALS. So historically, what we learn from literature is that the diversity of neurons and glial subtypes in the brain is astonishing. This is indeed a double-edged sword because on one hand, these diversity of neurons can map back to behavioral and all of our behavioral and cognitive function can be mapping back to different neuron subtypes in different cortical region of our brain. However, on the other hand, it is also the root of vulnerability and comprehensiveness of multiple neurological disorder, which is very hard to identify a drug target. So the central question in our lab that we've been always asking is that whether or not we can identify the cellular vulnerability to neurodegeneration in the human brain, and also do neurodegenerative disease converge on a shared cellular and molecular vulnerability, which opens an opportunity for us to identify a strategy that might target multiple neurodegenerative disorder. So today, as I said, I'm going to focus on ALS. ALS is a devastating motor neuron disease that can cause paralysis, but eventually death because of organ failure. And traditionally, what we learn is that many studies were primarily focusing on spinal cord. However, we should actually recognize the importance of the rest of nervous system that with a higher order. And the hallmark phenotype of ALS is indeed the massive cell death in the layer 5 upper motor neuron, namely BES cells, localized in what I'm highlighting here, the primary motor cortex. So the question to this project is mainly trying to ask which cortical cell subtypes are vulnerable in ALS, and can we actually identify shared vulnerability signatures in ALS. So before our paper is published, there are already a couple of groups, for instance, the Callis group and the Jashev group, their study have shown that from here, they try to link some of the GWAS, link the genes back to ALS, and not only did they find the enriched cell type from enriched molecular marker from the layer 5 upper motor neuron, but also they do find some of the markers that are expressed or enriched in the layer 3-5, which is upper motor neuron outside of layer 5. And here another study is showing that not only the layer 5, but other excitatory neuron from the upper motor layers show the high gene dysregulation in ALS. In brief, ALS is a very heterogenic disease with a very large spectrum. So to try to narrow down our target, we have tried to study only focusing on the C9orf72 gene that can cause ALS, and it is actually the most commonly found familial mutation that can cause ALS in European population. And the cause of the ALS pathology is mainly due to the repeats that you can find from the C9orf72 genes. Together, we have tried to strategically choose the primary motor cortex region that is known to govern the upper limb to narrow down the region that will cause ALS. And we have sampled and performed a single nuclei sequencing. And our data set is spanning 20 C9HRE patients together with 8 healthy brain control. So as shown here, the result of the single nuclei data set, we have successfully integrated the single nuclei collected from the 28 donors and managed to compile a single nuclei atlas with 360,000 nuclei. And they are high quality that enable us to identify 72 cell subtypes from the atlas, which includes some of the excitatory and inhibitory neurons, but also non-neuron subtypes, including the oligodendrocyte, microglia, astrocytes, and OPCs. So, commonly agreed cell type vulnerability in the field is mainly the decrease of cell abundance due to cell death and also high gene perturbation, meaning that there is a high number of differential expressed genes that you can identify from ALS comparing to control. What we are showing here is, again, we have seen that the layer 5 hallmark cell type is identified with the vulnerability. But what's intriguing is that we also identified 2 of the upper motor neuron subtypes that is coming out beyond layer 5, layer 2, 3 subtypes that also show the vulnerability that we identified. Although we acknowledge that there are also increase of cell type abundance on the right-hand side, we're still trying to find out what are the reasons behind that. So right now, after performing DE gene, we really want to understand what are the co-regulated gene profile among these vulnerability cell types. So what we have done is try to adopt this multicellular framework as developed in Julio's lab. Julio has just moved into our genome campus, and this is a very scalable tool that enables us to perform factorization method in order to find co-regulated genes in all of these subtypes without sacrificing the heterogeneity of the donors. And it is building on top of the MOFA framework. So after we have performed multicellular on our single nuclei data set, what you're seeing here is the 5 factors representing different gene programs, and each of the column is basically showing the correlation of these gene program to each of the sample. And these samples are coming out from disease and control. And usually what we will look at is the first factor because it basically representing the most important factor that might suggest the biological consequences caused by the gene. And what you can see is that after I've done a 1D clustering, the correlation of the first factor indeed show a strong correlation that can drive the ALS separation out from the control. As mentioned at the very beginning, ALS is a very heterogenic disease, so it is not surprised that not only we see disease is separated from the control, but the subset of ALS with frontotemporal dementia, it shows a heterogenic correlation between ALS and control as well, which we found very interesting. So with MOVA-cell, we can now look into each of these factors. For here, I've chosen factor 1, and to really look into the best explained cell types that drive the differences in factor 1. What we are seeing here is that if we try to subset if you try to give a threshold to the explained variant subtype, once again, not only we're seeing layer 5 excitatory neuron in this factor 1, but also we're seeing different excitatory neuron coming out from upper motor neuron, layer 2, 3 ITs, and also some of the inhibitory cell subtypes. And right now, when we try to subset the gene sets into only focusing on these best explained cell type, and to look into the correlation of the gene programs to these cell type, we found that there are a subset of upregulated gene, which is colored in red in this heat map. So we try to set a very stringent threshold, which these genes are all upregulated in all of these cells, and performed a graph-based analysis as shown on here. Not very surprised that we see pathways correspond to these genes are coming out from ribosome, translation, mitochondrial, and cell death, because you can imagine that these cells are really stressful while they're in the ALS disease pathology. But very interestingly, we also found that these gene sets are coming out from proteasome system and VAT-PACE system. With these gene sets, we compiled and correlate them into different pathways and perform a basic gene score analysis. And once again, if we focus in VAT-PACE and proteasome system, not only, again, we're seeing this is enriched in the layer 5 best cells, but also some of the cell type of interest coming out from layer 2, 3. And with a more sophisticated statistical framework, we run a KEC pathway analysis, and we see some of the significance pathway coming out are also associated to these two pathways. So in brief, the VAT-PACE is actually a receptor on an organelle of the cell that is called lysosome. And the role of VAT-PACE is try to pump proton out from the lysosome in order to maintain the pH inside lysosome. And lysosome is the organelle that is responsible for protein degradation. So we try to make a hypothesis that actually the VAT-PACE system is dysregulated in the neuron that cause the inefficient of protein degradation, especially one of the phenotype of ALS is protein aggregation as if Alzheimer's disease. And because of the inefficient of the protein degradation, that leads to the consequences of ALS. So with that, I'm going to talk about some of the future work that we have already tried to integrate multiple neurodegenerative disease atlas together in order to really find out whether these pathways are either specific to ALS or actually commonly shared among multiple neurological disorder. And another thing we have been doing is that we're trying to identify gene sets to derive patient-derived candidate genes guided in vitro functional assays for the detection of ALS in collaboration with Andrew Bassett and also with a very talented PhD in our lab, Matthew. So with that, I really want to take this opportunity to thank the PhD student, postdoc now, Eugene, coming from Singapore, to kickstart and also do some of the early analysis of this project. I would also like to thank the wet lab people, Fanny, Kenny, Ollie, and also Tanya for generating such an amazing data set. And also thank the very talented clinical PhD, Matthew, for doing some of the pathway analysis with me. We would also like to thank our collaborator, Andres Lakatos, and also my actually first supervisor, Martin Hamburg, for supervising this project with Omer Bayraktar, my supervisor, and also Jamin for providing for providing the samples from King's College London. Last but not least, I would like to thank the donor and also CCI and Wellcome Trust. Yeah. One last thing is that After I've convinced you about we have found shared vulnerability in neurons, I would also like to remind you not to forget to have the shared local favorite food, 茶餐廳奶茶蛋撻, after the HCA conference. Thank you very much.
Thank you, Jimmy. I'm sorry, I think we have to keep the discussions for the break. I mean, I have a lot of questions to you. I'm very interested in ALS. So sorry for going over time, but without further ado, I'm handing over to the fireside talks. So thank you very much for following this session.
Hello, everyone. Good morning. So yesterday, we started with a series of lightning talks. To remind you, 80 abstracts were submitted to be considered for oral presentations. 12 of them were selected for lightning talks. Yesterday, we heard 6 of them. Today, we'll be hearing the remaining 6 of them. Each speaker will have 3 minutes. I already see 4 speakers here. I would request everyone to come in front. This session is going to go super fast. And in case you have forgotten me, I am Dr. Shejruti Shaha from the Child Health Research Foundation in Bangladesh, and I'll continue moderating this session also. We will start with Dr. Shota Sasagawa. He's a research scientist at the Laboratory of Cancer Genomics in Riken in Japan.
Thank you very much. Thank you very much for your introduction. Sorry. Hi, everyone. My name is Shota Sasagawa. I'm a researcher in Dr. Nakagawa lab at Riken in Japan. Today, I would like to talk about spatial transcriptome and genomic of non-tumor disease tissues. So, advances in single-cell and spatial transcriptomic technologies have enabled detailed analysis of cell-specific changes and spatial gene expression patterns in disease tissues. In these studies, we focus on intracranial aneurysms and chronic hepatosis to analyze spatial gene expression and somatic mutations associated with these diseases. Our goal is identify new therapeutic targets, thereby contribute to the development of novel treatment for disease conditions. So first of all, aneurysm involves the weakness of blood vessel walls, so leading to bulging and the risk of ruptures. So we collected paired tissue and blood sample from human aneurysms along with clinical data and identified PDGFR-beta and other somatic mutation genes specific to aneurysms. So we biochemically and pathologically integrated how these aneurysms, somatic mutations contribute to aneurysm formations. So we used 10x Genomics vision platform and other platforms. Mouse models. So using vision data, we compared the expression level of various genes and found PDGFR-beta expression significantly increased with when mutation presents. Furthermore, gene related to MAP kinase and ARC pathways, as well as interferon genes were high expressions. So integrated these results finding, we identify the process of aneurysm formations. Next, we focus on chronic hepatosis. There are several causes of chronic hepatitis, but we detected group of virus infected cells and identified difference in immunosignal by comparing infected cells with their surround cells. So due to time constraints, I have provided a brief overview. So please come see our poster to learn more details. Thank you very much.
Thank you. Wonderful start. We got back 12 seconds. I'm going to welcome our second speaker now. Our second speaker is Mr. Hyungtai Sim, and he is a fifth-year graduate student in Seoul University. The floor is all yours.
Thank you for kind introduction. I'm Hyungtai Sim from Seoul National University, supervised by Dr. Murim Choi, and it's an honor to share our work at HCA Asia 2024. Today, I will talk about our preprint titled Systemic CD8 plus T-cell effector signature predicts prognosis of lung cancer immunotherapy. Patients' responses to immune checkpoint inhibitors are heterogeneous, and several contributing factors have been identified. In this study, we focused on the less explored interplay between genetic variants and transcriptomics of circulating immune cells. To understand this, we analyzed single-cell EQTLs and transcriptomic networks using data from 73 non-small cell lung cancer patients treated with ICI, combining single-cell RNA sequencing and genotyping via microarrays. Our analysis identifies 3,616 genes across 8 immune cell clusters, labeling cell type, specific, and treatment-dependent regulations. Comparing these with external data sets, we found 702 unique EQTLs initiated in disease-related pathways termed lung cancer EQTLs. Using multi-adaptive shrinkage, we explored cell type and treatment-specific gene regulations such as TNF and its receptor in monocytes and CD8 plus T cells. Co-localization analysis revealed association with immune and blood-related traits, including GSDMB, which showed pleiotropic associations to multiple autoimmune diseases. We constructed gene regulatory network to explain ICI responses. Among the WGCNA modules, the CD8 broad network, which is highly active in non-responders at the baseline, was linked to the AOMS TBX21 regulon, key to CD8 effector differentiation and exhaustion. Projecting this network onto tumor dataset revealed activation patterns in exhausted CD8 T cells. Additionally, activated CD8 plus T cell proportions explain the responses to ICI and survival outcomes. Lastly, we examined EQTLs within the network, finding enrichment in central genes. However, their effect sizes negatively correlated with centrality, suggesting broad regulatory loss even as core genes remain tightly non-genetically regulated. To summarize, our single-cell EQTL analysis revealed condition-specific regulation across immune cell clusters and treatments in lung cancer patients. We identified a transcriptomic network explaining prognosis can be extended to the tumor microenvironment. Lastly, genetic regulation via EQTL acts as a broad controller of gene expression networks. This concludes my talk. I thank the patient, collaborators, and lab members who supported this work. For more details, please see our preprint or find me at the poster session in 36. Thank you.
Thank you. We'll move on quickly to our third speaker. Our third speaker is Professor Bingji Chen, and she's from the Guangzhou Medical University, where she's a professor.
Hello, everyone. It's really my honor to present our recent work here, and I'm Bingji Chen from Guangzhou Medical University. As we all know that the post-mortem samples can be a very valuable resources for the human cell atlas. Actually, typically upon the death of the organism, the life ceases immediately and the organ lost their functionalities within hours, while the cells gradually degrading over time. Actually, we do not know when the whole body preserved in the low temperature and how the cells changed. And actually, when we have a collaborate project with the forensic department, and we found that after death and cold storage for 20 days, there was still half of the viable cells, which is quite surprising. Actually, we also verified this in the mouse models, and we found the similar results, and significantly proportion of the living cells can be preserved after nearly a couple of weeks. And then we're really curious about what kind of these cells can be alive and how they manage to do that. Then we send these cells for single-cell RNA-seq, and we found that this actually various of cell types can be alive. And actually, when we compared this the long postmortem interval samples with the freshly collected samples from the surgery, hospital surgery samples, actually they still looks good, and so it indicates that all the cells can preserve well. And we're also curious about how they manage to that. And when we do the gene differentiation expression analysis, and we find that they have a lot of changes in the stress adaptation and the environment adaptation. And actually, these changes is transient and reversible. And we also curious that these cells might be undergoing apoptosis or do not in a good conditions. So we try to culture these cells in vitro, and with success. And we found that we can culture these alive cells in vivo and into the organoid, and these cells can also be continuously passagized. And then when we compare the cells recovered from the fresh samples and also the post-mortem samples, and actually they do not seem quite different, and so which is really good. And we also want to use these alive cells, so we could culture some cells we regenerated from the young individuals, which these cells grows very fast. And then, to our surprise, the cells from the fast-growing individuals, and they can affect the organoids from the old or the diseased samples. And when we do the cell-cell communications analysis, and we found that the proliferation related pathways has been activated. So to sum up, and our result find that nearly after 20 days of the death, there are still actually nearly half of the viable cells, and these viable cells can grow into the organoids, and these regenerated organoids can promote the growth of the disease or the old organoids. Thank you. That's my presentations, and welcome to join our lab. And thanks for all my collaborations and all my students. Thank you.
Thanks, Mingji. Next, we will have Ms. Jingmeng Jia from Tsinghua University, where she is a research associate.
Okay, good morning, everyone. Today I will give my talk about the panvascular atherosclerosis. Atherosclerosis is a kind of disease and is a common causes of lots of fatal diseases, including cardiovascular diseases and cerebral vascular diseases. So in the main field of now, we mainly face the two questions of this field. The first one is, as atherosclerosis may affect the arterial systems throughout the whole body, so we want to know that what is the underlying common mechanisms of atherosclerosis in different arterial locations. And the second is, can we just obtain the data in the early stage of atherosclerosis in human samples? So based on these two questions, we designed a study to map the multi-cross samples from both human and mouse samples to generate the samples from different stages of atherosclerosis to investigate the differences of stable and unstable plaques. Based on these questions, in this research, we mainly focused on the MPCs, the mononuclear phagocytic cells of atherosclerosis. So we first characterize the functions and also the multiphased activations of MPCs in the early stage of atherosclerosis. And then, in the next step, we map the human samples, which represents for the late stage of atherosclerosis, to the early ones, which means in the mouse samples. And we want to recognize the common cells in both mouse and the human samples. And luckily, we found a subcluster of MPCs, which we gave a name of plaque instability monocytes and macrophages. And these cells can be exist in both human and mouse samples. And we also found that these cells is more abundant in acute coronary syndrome samples, which we include these samples as a reference of unstable plaques. And also, we conducted several experiments to validate this kind of research. In the panel C, we can see that in the virtual cell induction experiment, we have validated that these cells can be generated under the high concentration of oxidized low-density lipoproteins. And also, we can detect these cells for not only the tissues, but also detected in blood samples in human PBMCs, and we found that the high levels of these cells could be detected in the K samples compared to the normal controls, which means the markers of the cells could be a potential early diagnostic markers for the instability of atherosclerosis. So we also combined the spatial transcriptomics analysis data to investigate the transition of different regions of atherosclerotic plaques, especially not only the stable and unstable plaques, but also we want to know how the cell transitions between different states. For example, we want to know the differences between the unstable plaques before and after the plaque rupture. And also we detected several markers of the PIMs, which is markers of these cells which are specific to the plaque instability. We hope these cells could be a potential diagnostic markers for heart attack or other diseases like stroke. So in summary, in this study, we have built up a reference for the atherosclerosis, and we reviewed the distinct cellular compositions and gene expression patterns of both stable and unstable plaques. And also, we show the specific functions across different plaque regions. And finally, I want to thank my colleagues in Xuegong Lab of Tsinghua University for their support and collaborations. And if you have any questions or further interest in this research, you can feel free to contact us or find me in the poster session. Thank you.
Thank you. And now it is my pleasure to bring back on stage the moderator of the best breakout session from yesterday, which I, of course, co-moderated. So Kian Hong from Genome Institute of Singapore, who's a scientist there, will speak to us now.
Thank you very much, Juti. So good morning, everyone. My name is Kian Hong, and I'm presenting this lightning talk on behalf of the Asian Immune Diversity Atlas Network, or the AIDA network for short. So in this HCA Asia meeting, you've heard quite a bit about the lack of genetic and geographic diversity in genomics datasets, including in single-cell genomics datasets. So in the AIDA network, we've been able to harmonize experimental and computational workflows across our sites in India, Japan, Singapore, South Korea, and Thailand, where each of our teams actually performed 10x5'v2 single-cell experiments in their respective countries. So as a result, we've been able to go beyond continental level descriptors such as Asian to really look at the impact of subcontinental human diversity on immune cell phenotypes. So in our current phase of the atlas, we've got around 1.2 million single cells from 619 Asian donors, and you can access our CZ cell by gene visualization of the dataset via the QR code in the top right-hand corner of this slide. But in this lightning talk, I wanted to focus on two vignettes to highlight why it's important to study human diversity. So the first anecdote relates to cell type proportions, and we started by examining the incremental variants explained by covariates such as age, BMI, self-reported ethnicity, sex, or smoking on immune cell proportions in the AIDA Singapore cohort. So what we find is that we've observed multiple differences across our population groups. And one example I'm showing over here is the mate cells, which are higher in proportions in the Singapore Chinese donors and lower in proportions in the Singaporean Indian donors. Overall, we find that there's a substantial impact of sex, self-reported ethnicity, and genetic ancestry on immune cell proportions. The second anecdote I wanted to talk about relates to expression quantitative trait loci, or EQTLs for short. So what we did was that we looked at EQTLs in AIDA, as well as EQTLs in the GTEx version 8 whole blood dataset, which is around 85% European in the donor pool. And when we look at the EQTLs that are present in AIDA only, around 20% of them are actually low frequency to rare in the 1,000 genomes European superpopulation. So what this suggests to us is that there's an abundance of population-specific functional variants that can impact cell type-specific gene expression. And to really discover and understand these, you have to really leverage upon the full spectrum of human diversity. So if you come visit me and my poster, I'll be able to discuss how some of these other EQTLs can contextualize disease-associated GWAS loci, including Asian-specific variants. So in summary, we think that studying human diversity is a pressing equity issue and also of scientific importance. And I'd like to thank all IDA study participants as well as the entire IDA network. And it's truly been a privilege and pleasure to be able to collaborate with such fantastic, generous, and also very talented researchers across Asia. And I'd like to thank the CZI, Trans-Zacapa Initiative, Jonah Koo, and the Cell Science team for supporting our network. To thank all of you in the audience for listening, and I'm looking forward to seeing you at my poster. That's D214. So see you in a bit, and thank you.
Thank you. Last but not the least, now we have Ms. Jongha Lee from Seoul National University.
Thank you for the kind introduction. I'm Jongha Lee from Seoul National University, South Korea. Sorry for the long title, but what we did is quite clear. We performed single-cell EQTL of COVID patients from Korea cohort. We recruit approximately 200 individuals infected with COVID-19, exhibiting either mild or severe symptoms, and their blood samples were collected in longitudinal manner. Whole genome sequencing and HLA type were performed for genotype profile, while single-cell RNA sequencing and cytokine profile were conducted to characterize phenotype. By using 7 major cell types, we performed Söderberg single-cell EQTL analysis, and we identified interactive EQTLs, which are dependent on disease severity or time points. To evaluate our EQTL result, we leveraged GTEx whole blood samples and Japan COVID task force data, and we performed co-localizations analysis with GWAS data from host genetic initiative. And we also annotate genetic features on our EQTLs, and we observed that our EQTLs demonstrate the known genetic features, such as enrichment near transcript star size, enhancer, and 5' UTR. And by using HLA type as genotype profile, we perform HLA EQTL, and we observed that signals from HLA-B 1501, which is known to be enriched in asymptomatic patients. Similarly, cytokine profile serves as phenotype profile for pQTL, and we observed the higher heritability value for cytokine compared to RNA. To identify upstream regulator of severe-related IQTL, we proposed the model where difference in transcription activity influence DNA binding ability at the upstream of the target gene. Finally, we explored the predicted potential of genotype for disease severity, and we observed the model using IQTLs achieve AUPRC of 0.903, demonstrating strong performance. And our analysis were done with my colleague Eun-young Chung and Professor Morim Choi, and Liang Yu and Professor Siming Zhao from Tumors helped us a lot, especially in calling EQTLs, and thank you for your attention.
Thank you, and thanks to all the speakers. Can we get one round of applause again for all the speakers, please? So we are perfectly on time, and I think it's now time to break for refreshments, and we will be back after that. Thank you.